In class last Wednesday, on the day before fall break, we started to talk about ordered and unordered tetrads, and how looking at unordered tetrads and the order of spores within them, we can understand the orientation of chromatids during meiosis and whether recombination had taken place. So for example, in this example on the left, we have no crossing over occurring. So in this case, we have a dominant big A allele and a recessive little a allele. So during meiosis 1, the big A and little a alleles segregate from one another into two daughter cells. So this is referred to as first division segregation, because that's when the two alleles segregate. During meiosis 2, we then get the daughter cells produced, and in these types of fungi, there's an additional round of mitosis that occurs so that each meiotic product, each spore, actually produces two daughter spores. So as a result, in the final ascus, the ordered ascus, we have the following pattern of spores. We have 4 big A alleles and 4 little a alleles. So this 4:4 or 1:1 ratio is consistent with this M1 segregation pattern of no crossing over. In contrast, if crossing over occurs between the gene and the centromere shown here, segregation of big A and little a does not take place during meiosis 1. We could see that big A and little a are still present in each of these daughter cells. It isn't until meiosis 2 that big A and little a finally segregate away from one another into their own daughter cells. Following an additional round of mitosis, we end up with the following pattern of spores in the ascus, which is very different from the M1 segregation pattern. We have 2 big A, 2 little a, 2 big A, and 2 little a. So this deviation from that 1:1 or 4:4 ratio of the M1 pattern is consistent with M2 segregation and crossing over and recombination. If we look at these 4 spores in the middle, though, in this ascus, we can see that these are recombinants, and that they contain pieces of both the orange and the brown parental chromosomes. So in this example, 50% of the chromatids are recombinant. So we can use this recombination frequency, this 50% recombination frequency in this ascus, to calculate the distance between the gene and its centromere. So just to be clear, that the frequency of the M2 segregation pattern, frequency of the M2 segregation pattern can give us information about the distance from a gene to its centromere. But since only half of the spores in an M2 ascus, if you recall, half of the spores are recombinant, we have to take that into account when we calculate the recombination frequency. So to calculate the recombination frequency between a gene and its centromere, we can then figure out the number of M2 segregation patterns we observed, but we divide that by 2, right? Because only half of the spores are recombinant. So every M2 segregation pattern we see, we have to remember only 50% of the products are recombinant. We then divide that by the total number of asci we examine, and that gives us the recombination frequency between a particular gene and its centromere. So let's take a look at this example here. Here we have 6 different asci. We have the first one here and the 6th one here, and the spore order is reflected from top to bottom. So you can see we can go through these different asci and label them as being either M1 or M2. So in this first example for the 126, we could see there's 4 big A and 4 little a. So that would be an M1 segregation pattern. Similarly, for the 132, there's 132 asci that have this pattern, little a and big A. That would also be an M1 segregation pattern. But the remaining asci have very different patterns. Here we have 2 big A, 2 little a, 2 big A, 2 little a. So that would be M2 segregation pattern. Here we have little a, big A, little a, big A, which is also M2, and so on. We have big A, little a, and big A. Even though there are these 2 groups of little a together, we still don't see that 4:4 ratio that we see with M1. So this is also an M2 segregation pattern, as is this last example here. So if we wanted to calculate the distance between gene A and the centromere, we would simply calculate, so again, the recombination frequency, which would be the total number of M1 asci. So those are all of the ones that are M1, divided by 2, because only half of the spores in each one are recombinant, divided by the total, which in this case is 300 spores for asci, which would give us then a value of 0.07, which is 7%. So the distance between the gene and its centromere, in this case gene A, would be 7 centimorgans, or 7 map units. Shown on the bottom of the slide are the actual recombinant chromosomes, showing how they would segregate during meiosis. So again, during meiosis, right, the microtubules pull away from the centromeres. The sister chromatids are pulled away at the centromere. So this top chromatid would end up as the first spore. The second chromatid would end up as the second spore. This third chromatid ends up as the third spore. And this fourth chromatid ends up as the fourth spore. So the order of the spores then reflects the orientation of the chromosomes during meiosis. We can take a look at another example on this next slide, dealing with Neurospora. And a methionine auxotroph, which we'll indicate here with a lowercase m, crossed with a wild type, with a plus. So again, this would actually be a MET plus. This would be just a MET minus. But we'll use M and plus to indicate auxotroph and wild type. So from this cross then, we'd expect the cells, or the two haploids to fuse together. So a mutant crossed with a wild type. We would get a diploid cell that then goes through meiosis. And then just like in the previous example, we would end up with asci produced, different types of asci, depending on whether recombination takes place or not. So in the following example, we have these different classes. We have 6, 5, 6, 7, 40, and 36. And the spore order is reflected in the table from top to bottom. So rather than showing all 8 spores, this table is basically showing that each different genotype corresponds to 2 spores. So this would be, the first 2 spores would be wild type. The next 2 spores are mutant. The next 2 spores are wild type. The last 2 spores are mutant. So the question is asking how far is the gene from the centromere in this example. So we would just go through and determine which ones are M1 or M2. So these first 2 are M1 segregation pattern. We have the 1-to-1, right, or the 4-to-4 ratio of mutant to wild type. The remaining asci, though, are M2. These have the M2 segregation pattern. So again, to calculate the distance between the methionine gene and the centromere would simply be the M2 frequency over 2. So this would be 6 plus 5 plus 6 plus 7 divided by 2, since only half of those spores are recombinant, divided by the total, which was 100 in this case, which would give us 0.12, which is 12 centimorgans. So in this example, the centromere to methionine distance would be 12 map units. So we can maybe dig a little deeper in this question and ask, how did these different spore patterns become produced? Where would recombination have to occur to produce these different spore patterns? So let's take a look at this first example here, the spore pattern of a plus M. So we'll put a 6 here just so we can give ourselves some orientation. And we'll draw the two homologs here. So we have 2 homologs in the cell. Again, it's a diploid cell where we have the mutant allele on one pair and the wild type on the second pair of sister chromatids. So we want to ask ourselves, how would meiosis have to proceed to produce an ascus with this following pattern of spores? So in this case, we could predict then that crossing over takes place between the top chromatid and the bottom chromatid. Right, if that were the case, right, this first sister chromatid would be wild type, so that would be the first sister chromatid. The second sister chromatid is a non-crossover, so that would be the M. This third chromatid is also a non-crossover, so that would be wild type as well. And this fourth chromatid would be a crossover which would correspond to M. So there, a crossing over between those two sister chromatids would yield the following spore pattern. Let's take a look at the next one, this one that has the five different asci that had that particular pattern. So again, we can draw our sister chromatids. And again, it's a methionine mutant on one and a wild type on the other. And in this case, the spore pattern was M plus M. So again, where would crossing over have to occur for this spore pattern to be produced? And in this case, we could have crossing over between the second and the fourth sister chromatid. Right, so in this case, the first chromatid would be a non-crossover. The second chromatid would be a recombinant, right? So that would be the wild type. The third chromatid would also be a non-crossover, so a wild type. And this fourth chromatid would be a recombinant, yielding the M, or the mutant allele in the fourth spore. Just for thoroughness, let's go ahead and do the third one here, this third six class here. We could draw this one out again with the homologs with mutant M and wild type on the other pair. And in this case, we could have crossing over that occurs between the second and the third spores. Right, in this case then, the first chromatid would be non-crossover, which would be an M. Oops, that shouldn't be popping up. The second, so that would be an M as the first spore. The second chromatid is a crossover, right? That would give us the wild type. The third chromatid is the M, so that would be the third spore. And the last chromatid would be wild type, that would be the fourth spore. And lastly, I won't go through this one in detail. Let's just say for the seventh one, we could just draw this one out, and you can work through it on your own. The crossover would have to occur to produce this genotype between the first and the third chromatids, and that would then yield this plus M plus pattern. So we can actually draw out the homologs and predict where crossing over would occur to produce these spore patterns. And of course, there's always the complexity of multiple crossovers that go undetected, but we're not going to worry about that right now. We'll just assume, in this case, for a single crossover, the easiest, the most simplest explanation is sufficient to describe how these different spore patterns were produced. So we can go beyond just calculating the gene to centromere distance and actually map the distance between different genes in both ordered tetrads and unordered tetrads. And here's where we look at the genotypes of individual spores with respect to two linked genes. So let's just consider an example here where we have two haploid spores, one that is AB crossed with one that is recessive AB. So if these two spores are fused into a diploid, we would get the following genotype. Right? And the way this is written is that A and B, the genes A and B, are linked. Right? That's the way I've written the genotype to indicate that these genes are linked. If this diploid, so again this is a diploid cell, if it were then to go through meiosis, there are 4 different gametes or 4 different spores that could be produced. So let's outline the different examples here. So let's say, for example, that no crossing over occurs. If there's no recombination that occurs, and the cell goes through meiosis, right, there were 4 spores that would be produced. The parentals, they're both dominant for A and B, and the recessive for A and B. So in this case, right, the 4 spores that are produced are identical to the parents. So this is referred to, this type of ascus, so again, this is within a larger ascus here, these 4 spores. This is what's called a parental ditype. Well, let's call that PD for short. What that means is that there's 2 types of spores. Right? There's the big A, big B, little a, little b, and they're identical to the parent. So this is also consistent with non-crossover, no crossing over. So that's one outcome. Another possibility is where we have 4 spores but all 4 of them have a different genotype. So here we have all 4 possible combinations of alleles. So in this case, we have to ask how many different genotypes do we see in this ascus, where we have big A, big B, little a, big A, little a, big B. So this is called a tetratype because there are 4 different genotypes in the ascus. And we could just abbreviate that with a T. So these are often formed by a single crossover. So let's say single crossover underneath here. And just to illustrate that, if we were to draw out the homologs here, here's a pair of chromatids, and here's another pair of chromatids, and we have big A, big B, big A, big B, little a, little b, little a, little b. Right, a single crossover between those 2 chromatids would produce a big A, big B spore, big A, little b, big B, and little a, little b. So a single crossover is all that's required to produce these 4 different types of spores. Lastly, the third type of spore we could observe is one where we have 2 genotypes. So little a, big B, little a, big B, big A, little b, and big A, little b, all contained within an ascus. In this case, there's only 2 genotypes, right? So we would still call this a ditype, but these are different from the parents. So this is called a non-parental ditype, or NPD for short. And these arise through a double crossover. So the rarest, if you're calling it, double crossovers are always the rarest events. And here's another example of maybe the rarest spore type we would expect to see. So in this case, if we draw out our 2 homologs, we have big A, big B, big A, big B, little a, little b, little a, little b. If crossing over occurred here between those 2 non-sister chromatids and between these 2 non-sister chromatids, that would yield, kind of run out of the room here, so I'll draw the spore pattern over here. That would yield a big A, little b. So we go from, I can draw it just to outline this. The first spore would be big A, little b. The second spore would be big A, oops. Yeah, big A, that's right. I thought I was doing a tetratype. Big A, little b. The third spore would be little a, big B. Let me get that in blue so we can see that. Right, little a, big B. And similarly, the last spore would be little a, big B. So in this case, there's 2 crossovers that have occurred between A and B. We have 1 crossover that occurred here, and 1 crossover occurred there. So that's a very rare event, that there would be 2 crossovers between those genes, and therefore the non-parental ditype would be produced very infrequently, if at all. So again, that could be used as an indicator that the genes are either very close together or very far apart. So this is where we're going to stop for this online lecture for today. And I'm hoping you understand the basic idea between how these 3 different spore types are produced, or different ascus types are produced: parental ditype, tetratype, and non-parental ditype. And in class on Wednesday, we're going to go on to the next slide, which kind of highlights some examples of how these different asci are produced, and then talk about how we can use that information to calculate gene-to-gene distances in fungi that produce both ordered tetrads and unordered tetrads. So if you have any questions, feel free to email me, or I'll be back on campus on Tuesday morning. And otherwise, I'll see you all in class on Wednesday. I hope you all have or had a good break, and we'll see you soon.